This is our final presentation of our project Antibacterial Defensive Spices by Yitian and Nick Matisse.
For our introduction to background research, we found that spices were often used as a means of medicine in the past due to their natural antibacterial properties, and even for jobs like masking the scent of unpleasant odors or keeping food from spoiling. The relevance of testing spices' not antibacterial capabilities is an important factor to understand the possibilities of disease prevention and curing. The purpose of this experiment is to test the antibacterial properties of different spices to see how they react to and counter E. coli.
Our hypothesis was if oregano, thyme, cinnamon, onion powder, and salt were to be put in petri dishes with E. coli spread on them for 48 hours, then cinnamon will create the largest inhibition zone because it contains essential oils that counter E. coli and many other bacteria. And this was our source for that hypothesis.
For procedure, we had sample sizes with 2 petri dishes, 1 swab of E. coli, and a half a teaspoon of spices. The test subjects were the spices, which were oregano, thyme, cinnamon, onion powder, and salt. The independent variable were the spices. The dependent variable is the size of the inhibition zone in millimeters. The constants were the petri dishes, the amount of each spice, the time given for each spice to take effect, and the attempted even spread of bacteria in the petri dishes. The negative control is the petri dish with E. coli and no spice. There were 2 replicates, and data was recorded 48 hours from when the spices were added. And statistical analysis was through standard deviation and standard error, and each person carries out 1 trial.
Because we are handling E. coli, there is also a safety procedure. During this experiment, it is recommended that the participants wear safety gloves and goggles. The petri dish should be streaked evenly without touching the Q-tip anywhere besides the inside of the plate. The plate should remain closed until the end of the experiment once the E. coli has been added to the plates. For disposal, 99% isopropyl alcohol should be used to disinfect the plate and the swabs. For the plates, using gloves, the alcohol should be poured in and left to sit for 15 minutes to finish disinfecting. These can now be placed in the trash. The swab should also be disinfected using alcohol.
Figure 1 was replicate 1 after 24 hours, and there was little to no E. coli after 24 hours. In figure 2, it was replicate 2 after 24 hours, and there was also little trace of E. coli.
Here are more figures. This was the onion powder after 48 hours, and you can see it has a inhibition zone with visible areas of E. coli being inhibited. This is cinnamon. You can see the visible streaks of bacteria with the inhibition zone. This is thyme and oregano after 48 hours. There are streaks of bacteria, but if there is an inhibition zone, it is very difficult to see, and there's not one because of the spice bleeding into the petri dish around it.
This was our bar graph and annotation. After the 48 hours, it was found that there was a statistical insignificance between the cinnamon powder and the onion powder. Additionally, the thyme, oregano, and salt also showed to have a statistical insignificance shown by the error bars in the graph. On average, the onion powder performed 12 millimeters better than the thyme and 12.5 millimeters better than the oregano and salt. The cinnamon also performed, on average, 12.5 millimeters better than the thyme and 13 millimeters better than both the salt and oregano. Standard deviation and error were calculated using Excel.
This is our table with the annotation. So our table is the effect of spices on the inhibition of E. coli. And the annotation is after 48 hours, the cinnamon and onion powder showed to be statistically significant from the thyme, oregano, and the salt. The spice that contained the least variations of data was the onion powder with an average of 0.5 millimeters of air. The spice that contained the largest variation was the cinnamon with an average of 2 millimeters of air. On average, the cinnamon performed 1.5 millimeters worse than the onion powder, 1 millimeter worse than the oregano and salt, and 0.5 millimeters worse than the thyme. The onion powder performed an average of 1.5 millimeters better than the cinnamon, 0.5 millimeters better than the oregano and salt, and 1 millimeter better than the thyme. Standard deviation and standard error were calculated using Excel.
This is our summarized discussion. This is for our research. When a bacteria is being inhibited, it is unable to grow due to the protein denaturation from the inhibitor, where it then creates a circle around the substance. It was also found that cinnamon contains eugenol, a chemical compound that was tested to combat bacteria like E. coli. Eugenol acts as an antibacterial agent by disrupting the cellular membrane and killing the E. coli. Quercetin is a chemical compound found within onions. Through several experiments, it was found that it had a good inhibitory effect on the growth of many different bacteria, including E. coli.
Okay. The final claim of our discussion was cinnamon, which contains compounds like eugenol and carophyllene, would cause the largest zone of inhibition.
This is the summarized errors in our discussion. Differences in brand of spices could mean different recipes for each spice, which leads to different ingredients. This might skew the results as the antibacterial compounds could be different in different concentrations. The temperature is likely another factor of deviation because E. coli prefers a certain temperature, usually warmer temperatures. The temperatures were variable during the experiment, and the house temperatures between each of our houses would have led to further differences.
There were several future experiments that could be tested. During our experiment, we tested a fairly small range of spices. So testing a wider variety of spices would lead to more understanding of this topic. A related topic would be testing plants for antifungal properties. I believe peppermint was a plant in our research that has shown antifungal properties, so conducting more tests of that would be interesting. And finally, comparing effectiveness of the spices in this experiment of their extract oil versus the ground form of the spice.
This was the summarized relevance in our discussion. The relevance of this lab is that spices can potentially be used as cheap healthcare, as antibiotics can be expensive.
And finally, this is our reference section. So we had many sources that we used to finalize our ideas. So I'm going to pan through these. All right, that is the end of our presentation.
Thank you for watching.